In the sun-blasted cliffs overlooking the Dead Sea, a secret lay hidden for nearly 2,000 years. Then, in 1947, chance intervened. A young Bedouin shepherd, searching for a stray goat, hurled a stone into a dark cave and heard the unmistakable sound of shattering pottery. What he and his companions would soon uncover in that cave and 10 others nearby would become one of the most extraordinary archaeological discoveries of the 20th century, the Dead Sea Scrolls. This was no ordinary find. The caves contained a vast library, nearly 1,000 ancient manuscripts, preserving the oldest surviving copies of the Hebrew Bible, alongside a remarkable collection of religious, legal, and philosophical texts previously unknown to history. Written between the 3rd century BCE and the 1st century CE, these scrolls opened an unprecedented window into a pivotal era, a time that witnessed the transformation of ancient Judaism and the emergence of Christianity. Yet the discovery also posed a monumental challenge. Over centuries of decay, the scrolls had fragmented into more than 25,000 pieces, some no larger than a postage stamp. Reassembling them was like attempting to solve a puzzle with most of the pieces missing. From the very beginning, scholars debated their origin. Were they written by a small, isolated sect living at Qumran? Or did they represent the work of multiple Jewish communities across ancient Judea? For decades, these questions remained unresolved, until now. Before we continue, take a moment to subscribe and turn on notifications so you never miss the discoveries, stories, and revelations that are reshaping our understanding of human history. A revolutionary approach, extracting and analyzing ancient animal DNA from the parchment itself, is finally shedding light on where the scrolls came from, who created them, and how ideas moved across the ancient world 2,000 years ago. Discovered between 1947 and the 1960s in the 11 Qumran caves and other desert sites, such as Masada, the Dead Sea Scrolls are written primarily in Hebrew, with portions in Aramaic and Greek. They come from a time when Greek culture deeply influenced Jewish life, giving rise to diverse groups such as the Essenes, Pharisees, and Sadducees. Sorting the fragments has always been painstaking work. Scholars rely on handwriting styles, linguistic patterns, and textual parallels to link pieces together. Still, crucial questions persisted, especially regarding provenance. Where were these texts actually written, and by whom? The breakthrough began with a simple question: what were the scrolls made of? The vast majority were written on parchment, a durable material crafted from animal skin. For molecular biologists, this was the key. Every piece of parchment, no matter how small, carries a molecular fingerprint, traces of the DNA of the animal it once belonged to. Extracting DNA from 2,000-year-old parchment, however, is extraordinarily difficult. Ancient DNA is fragile, broken down by heat, time, and environmental exposure. It is also easily contaminated by modern human DNA from centuries of handling. To overcome this, researchers examined 39 ancient objects, including 26 scroll fragments, along with sandals, water skins, and garments recovered from desert sites. To avoid damaging these priceless artifacts, conservators collected microscopic samples, sometimes no more than dust, from blank areas of the parchment. Modern parchments were used as controls. The work was conducted in a sterile laboratory in Sweden, equipped with airlocks and ultraviolet light to eliminate contamination. Using a technique called silica-based extraction, scientists were able to capture fragments of ancient DNA like iron filings drawn to a magnet. The first task was identifying the animal species. DNA from the scrolls was compared to genetic signatures of animals common to the ancient Near East: sheep, goats, and cattle. The results were striking. Of the 26 scroll fragments analyzed, 24 were made from sheepskin, but 2 fragments, both from the book of Jeremiah, were written on cowhide. In the context of the Judean desert, this was explosive. The arid landscape around Qumran could support sheep and goats, but cattle require abundant water and pasture. There is no archaeological evidence that cows were raised or processed at Qumran. The conclusion was unavoidable. These Jeremiah scrolls were produced elsewhere, most likely in the fertile Jordan Valley, and later transported to Qumran. This single discovery dismantles the long-held notion that Qumran was an isolated sect producing all its own writings. Instead, it reveals Qumran as something far more dynamic, a hub of learning, a repository where texts from diverse regions and traditions were gathered, studied, and preserved. With animal species identified, researchers turned to a more ambitious goal: using DNA to sort fragments. Ancient scribes typically used the skin of a single animal for each manuscript. That meant fragments made from genetically related animals were likely part of the same scroll. Both mitochondrial DNA, passed down through maternal lines, and nuclear DNA, passed from both parents, were analyzed. Sheep populations can be grouped into maternal lineages, known as haplogroups, labeled A through E. Haplogroups A, B, and C were common in the ancient Near East.
The team focused on a mysterious non-biblical text known as the Songs of the Sabbath Sacrifice, a mystical liturgical composition. Multiple copies of this work were found in the Qumran caves, suggesting it played an important role in the community's spiritual life. But another copy was also discovered more than 50 kilometers south at Masada, the desert fortress. This raised a crucial question. Did the text originate at Qumran and spread outward, or was it widely shared among Jewish communities? DNA delivered the answer. All Qumran copies of the songs were written on parchment from genetically related sheep belonging to haplogroup B, indicating they were produced locally, likely from the same flock, by the same scribal community. The Masada fragment, however, came from a sheep belonging to haplogroup A, genetically unrelated to the Qumran animals. In other words, Masada did not receive a scroll from Qumran. Its community created its own copy. The text was not the exclusive property of a single sect. It circulated widely among different Jewish groups. The final challenge involved provenance. Many scroll fragments were not excavated under controlled conditions, but acquired from antiquities dealers in the mid-20th century. Their true origins were often uncertain. Some were assigned to Qumran simply because they appeared in collections associated with the caves. One fragment, in particular, 4Q344, had long puzzled scholars. Unlike most Qumran texts, it is a mundane legal document acknowledging a financial debt. Its script and later date also set it apart. Genetic analysis confirmed suspicions. Its sheep DNA did not match the main Qumran cluster. It was a clear genetic outlier. This provided the first hard physical evidence that the fragment originated at a different site in the Judean desert, likely a settlement where everyday legal documents were common, and was later mistakenly grouped with the Qumran scrolls. A fragment of Isaiah showed similar results, suggesting yet another unidentified origin. These findings demonstrate that some scrolls were misattributed due to their modern acquisition history rather than ancient reality. The implications are profound. For the first time, decades-old debates about the Dead Sea Scrolls are being resolved with independent scientific evidence. DNA has become an impartial judge, cutting through speculation and tradition. The scrolls are no longer seen as the product of a single isolated community, but as a reflection of a rich, interconnected world, where ideas,
texts and traditions flowed between groups during the Hellenistic and Roman periods. Some scrolls were written locally at Qumran, following distinctive scribal practices. Others were brought from distant regions, carrying alternative versions of sacred texts and new theological perspectives. The diversity of thought preserved in the scrolls mirrors the genetic diversity of the animals whose skins carried their words. And the story is far from over. As research continues, each fragment still holds secrets waiting to be uncovered, connecting the scrolls to the people who wrote them and to the wider world they inhabited. The Dead Sea Scrolls continue to speak, not just through ink and parchment, but through the very molecules of history itself. Thanks for watching. If you found this video insightful, consider giving it a like, sharing it with fellow history enthusiasts, and subscribing for more stories from the ancient world. Let us know your thoughts in the comments, and we'll see you in the next one.